Thank you for the introduction, and thanks for the opportunity to demonstrate some of the amazing work we're doing between a collaboration between our research at Birmingham and Liz Sockett's group at Nottingham. So the title's called Getting a Grip, the Molecular Mechanisms of Bdellovibrio Attachment. So this is a story about how we found a family of proteins in Bdellovibrio, and we managed to structurally characterize them. And it's quite a fascinating story. So Bdellovibrio itself is a fascinating organism. It's a bacterium that actually preys on other bacteria, and it's free swimming, and it swims around until it meets its prey, the other bacterium, and it attaches to the surface and then invades into its periplasm and then consumes the bacteria from within. So once it invades, what you can see is this delaplast forms, which is where the prey cell rounds up. And if you look at the little video on the right hand side, you can see that there's one that's already been invaded, and the one just underneath, as the video starts, you'll see that it gets invaded. So you can see the little Bdellovibrio there attaches and invades, and then you should see the one on the top left lies and release the progeny bacteria, and then the second one will pop. So this process takes about 3 or 4 hours. So it's just a really fascinating organism to work on, and it has so many applications for future therapeutics and biological control. So understanding the fundamental biology is really important to us. So this project started when Liz Sockett's group at Nottingham, they were working on a completely different project, and they were looking at this CPOB homologue in Bdellovibrio that's sort of involved in cell wall biogenesis of Bdellovibrio. And what they noticed is that when they fluorescently tagged this protein, the invaded Bdello, which you can see in blue inside the prey bacterium in the microscopy images there, it deposits this protein in what seems to be some sort of vesicle. And on the right hand side there, you can see some ghosts of the prey cell, which is just a sort of outer cell wall structure that's left behind in certain cases by Bdellovibrio. And you can see that this vesicle actually gets left behind, and it contains a wealth of predatory involved proteins. And they found this out because they managed to actually purify these vesicles to a high enough concentration where they could then do proteomics analysis. And that's where we actually identified, among many other proteins, a subset of the MAT family fibers that I'm going to be talking about today. So when you take the identified fibers that we got from the vesicle, and then you actually use BLAST for sequence homology, you actually find that there's many proteins, and they can be divided into sort of 2 groups. The first group is on the left. It's called the S74 protease proteins, and they're characterized by having a C-terminal S74 domain, which is an autoproteolytic domain that actually cleaves off itself and leaves a mature fiber at the end. And then there's this group on the right-hand side that actually don't have the S74 and have other domains at the C-terminus. So what's really interesting is that Bdellovibrio actually has a sort of analogous lifestyle to phage, in that it attaches to its target bacterium, and then although the life cycle is slightly different after that, they have to replicate within the bacterium. But Bdellovibrio seems to have these phage-like fibers, because the phage-like fibers on phage have these S74 domains. So you can see there on the right-hand side, when the protein is first expressed, you get this beta helix, which is characterized by these beta sheets that rotate around. And then you can see in the surface view the S74 domain. So that actually releases to form a native fiber that is adhesive to typically LPS on the surface of bacteria. And that's what you can see on the left-hand side there, the native fiber. So they took a subset of the fibers from Bdellovibrio, and they managed to do some fluorescence assays to see where these proteins localize. What's really quite interesting is that actually each fiber seems to have a different focus of fluorescence, indicating that it has different localization in Bdellovibrio. And as you can see there on the very left-hand side, you can see that actually the flagellum has a focus fluorescence for BD1334. And then what I would have thought would be more the case would be you'd get these fibers at the invading pole, which is like the front end of Bdellovibrio, where the opposite side of the flagella. So that's what you can see for 2439. But it indicates that the different fibers may have different roles in Bdellovibrio, but they're all likely to be adhesive in function. So when we actually managed to express a full length fiber, which this is one that we actually got from Twist. It's one that we couldn't claim because it was such a big gene. You can actually purify them and see them with cryo-EM. So that's what you can see in that image on the right-hand side. So you can see these fibers are actually quite flexible. So the first one we actually managed to structurally characterize was this S74 protease, BD3182. And what you can see on the SDS page is that if you express it in its native form, it actually cleaves. So that proved that it was a true S74 protease. And you can see the cleavage product of the S74 chaperone and the beta helix. And then if you mutate out the catalytic serine, you can see that actually you don't get that cleavage and you get the uncleaved fiber at a much higher molecular weight. So this video will show you the structure of BD3182, and you can see that it's similar in that it's made of 3 polypeptide chains that form that rotating beta sheet. And you can see that beta sheet lights up blue now. And this forms a cleft that we believe to be the ligand binding site. It's likely that the binding site is LPS, but we still have to find the ligand. So what you can see inside these proteins is really quite beautiful that you get this 3-fold symmetry in the hydrophobic core. It's not particularly functional, it's just really nice to see. So next, we've got BD2740, which is a fiber at the C-terminal end has a C-type lectin. These C-type lectins sometimes bind calcium. We don't believe this is a calcium binding lectin, and it may or may not actually bind sugars, which is what lectins typically do. So we still have to work on finding the ligand for that. We also got the structure of the C-terminal portions of 2 proteins that are very similar in their N-terminal portions of the gene, but actually are completely mosaic and swap out those C-terminal gene domains for completely novel folds. So typically, structurally, we'd take a domain and we can search through databases to identify other domains that have similar functions and similar ligands, if that's what we're looking for. But unfortunately, because these are novel folds, we don't have that ability. So as yet, we don't actually know what the ligand is for these. But with BD1334, we get a little bit of a clue from the crystal structure because in this little pocket formed in a cleft between 2 of the protomers of the trimer, you actually see a phosphate molecule. So it could indicate that this is actually a specialized fiber for binding maybe the phosphate groups of phospholipids or the lipid A of LPS, which is on the surface of other bacteria. And then finally, we managed to characterize the non-S74 TNF-like domain. So both of these proteins, although don't seem to have much structure, sequence homology, they do show structural homology in that they both have a C-terminal TNF domain. Now, TNF domains actually bind various ligands again. Some bind sugars, some bind proteins. But what we actually managed to do, and which is going to be the most with the focus for the rest of the talk, is BD2439, because we actually managed to characterize what the ligands were for this protein. So the first thing we did is we took the protein and we used a thermal unfolding assay to actually look at the melting point of this protein. And this is really useful because if you can get a ligand that binds to a protein, quite often it will stabilize it in a way that you can detect that in the fact that it unfolds much later. So when you unfold this protein, you get 2 transitions. You get this first transition, which is highlighted in blue, and we believe that to be the beta helix domain. And then you get a second transition where the C-terminal domain unfolds. And that's the one that we can actually see a shift in when we incubate the protein with different sugars. And what's really interesting is the sugars that actually seem to bind and increase the melting temperature are any where you get what's called equatorial and vicinal hydroxyls. So that's any hydroxyl groups from the sugar that point out of the plane and are next to each other. So that's, you could, for all these sugars, you can see that they all have these equatorial hydroxyls. And then if you take N-acetylglucosamine, it's got a pair of hydroxyls. If you take galactose, you can see that it's got 3 hydroxyls together. But then if you take one of those hydroxyls away in the middle and replace it for an N-acetyl group, you can actually see that it then loses binding. So that's really showing that it's important to have those 2 hydroxyls together. So we wanted to use structural methods to see these interactions and to see how this sugar interacts with BD2439. And I tried many of the sugars that you could see in that assay previous, but we actually only managed to get one high resolution structure of a sugar that was bound into the binding pocket of BD2439. And that was this probably non-physiological sugar on the right-hand side, which is actually a disaccharide found in peptidoglycan, which is unlikely to be its target molecule. But nonetheless, it actually acted as a really good proxy for how the sugars bind. So I'll show you this video now. So you can see there that it's got that beta helix as most of the other fibers do. And you've got the C-terminal domains at the end, they're the TNF domains. And when we focus in, we can see that when we soak the crystal in a cryoprotectant ethylene glycol, that binds into a pocket. And that gave us a really good idea of where this pocket was. And the pocket is formed by these charged arginine and histidine residues primarily. And what you can see is that the sugar that we managed to crystallize actually binds into the same pocket. And each fiber, because the pocket is between the chains, it actually has 3 binding sites for each of these sugars, which we saw completely occupied in all cases. So that's a close-up of the actual binding pocket with the sugar in. We could only model the one part of the sugar, the one, the actual Glucknack moiety of the disaccharide, because the Muirnack moiety was actually a bit too flexible to model. But you can see that it's a really nice binding pocket for those 2 hydroxyls pointing in and making many, many polar interactions with the residue side chains. So we now know that it binds sugars, but what we wanted to see was, could it bind to the surface molecules on bacteria, which is what we predict these things to do. We predict them to be fibers that are adhesive to the prey of Bdellovibrio. So we took the protein and subjected it to a fluorescence assay, which is called a glycan array, where you fluorescently label the protein, you put it on a chip where you've got all of these pathogenic LPS molecules labeled onto the surface. And LPS is actually the surface molecule that is buried into the membrane of bacteria. It's basically composed of a lipid that anchors it into the membrane, and then a long chain of sugars. So when we actually did the assay, what you could see is BD2439 actually bound to many of these pathogenic bacterial LPSs, which is really interesting because these organisms aren't actually ever seen by the strain of Bdellovibrio that we're actually using, because Bdellovibrio is found in the soil. So it's quite fascinating that it's got this redundancy to be able to adhere to epitopes of LPS molecules that it doesn't even see in its typical life. So then you can then take the O antigen of Proteus mirabilis, which is what it binds strongest to. And you can see that it's got many of these sugars where you've got vicinal hydroxyls. So it's likely that these sugars will actually be binding into the binding site that we saw the sugars in the crystal structure. But more importantly, there must be other interaction sites on that protein that give specificity to this interaction, because there are likely to be many vicinal hydroxyls in these other LPS molecules, especially the ones that don't bind. And for some reason, they're prevented from binding, but there's, even with the ones that bind here, you get obvious preference for Proteus mirabilis over other LPSs. So Proteus mirabilis itself is quite an interesting organism because it causes UTIs, and that's mediated by this hyperflagellation and swarming activity. So it actually differentiates from this bacterium on the left into this hyperflagellated bacterium on the right hand side, you can see there, and that allows it to swarm very quickly and actually colonize catheters. So what we hope is that we can actually utilize this protein for further diagnostic or therapeutic use, but that is actually a long way off right now. So we can also use modern methods to actually model the rest of the fibers as well. So what you can see here is the 6 fibers that I've demonstrated that we got crystal structures at the C-terminal portion. That's the region on the right, which we think is the prey binding end. But you can actually see that they're actually formed quite repeatedly by this C-terminal domain, and then an interspersing fiber, and then what seems to be quite a similar N-terminal domain, which is the domain that we believe to be binding to Bdellovibrio. So in summary, Bdellovibrio invades its prey bacteria and then deposits a vesicle which contains a wealth of predation-associated genes. And this vesicle, we can see that it deposits these fibers, and it's quite possible that it actually deposits this vesicle to remove the machinery involved in invasion prior to growth inside the cell. And these fibers that we identified bind to surface molecules on the prey. So we believe these to be important in the invasion process. And we believe that they can be utilized in the future to be of therapeutic or diagnostic use. Thank you for listening. And I'd like to thank everyone in the Lovering group and the Socket group over at Nottingham. And thanks again to Twist for inviting me to talk today.
Okay, thank you so much, Simon. It's been great. Okay, so I want to remind everyone who is on the call, it's on the webinar, that they can plug the questions on the on the chat box. So we have a couple of questions. Okay, so do these proteins produce downstream signaling, or they are just used for adhesion? Sorry, I don't know how to pronounce that.
Yeah, so it's a bit hard to say at the moment because we're still working. We've made quite a lot of work on understanding how it actually interacts with Bdellovibrio itself, as though it wasn't part of this talk. But I believe that these proteins are primarily just adhesive. I think the actual ability of what we can see to cause downstream signaling is minimal and quite unlikely with what we believe to be present. So I think these are for adhesion, and there's probably other proteins on the surface of Bdellovibrio that we haven't characterized yet that are actually involved in signaling after adhesion has occurred, because there must be something that signals for Bdellovibrio to attach and then to commit to the actual invasion.
Okay, perfect. We just got another question. Are you trying to get structural information of the rest of the fiber? For example, the N-terminal region?
Yeah, so that's what we've made quite a lot of headway with that right now. So yeah, the interspersing fibers I think will prove very, very difficult to structurally characterize. We can see from the EM that they're so flexible that would be hard to use electron microscopy to do that. And I think they'd also be far too flexible to allow us to get really good X-ray crystallographic data from as well. But yeah, for the actual N-terminal domain, we've made quite a lot of leeway on that so far. But again, I don't want to go into that too much today.
Okay, perfect. Thank you. Are these fibers found in other bacteria than Bdellovibrio bacteriovirus?
Yes. So again, we're currently looking at these. So what we've managed to see is that these fibers seem to be quite universal in other bacteria, and the actual diversity of these fibers is quite immense. So we see over 20 fibers in Dello, most of which have this typical beta helix. And then we see many with additional fibers, these non-S74s, but in other bacteria, you get a huge range of C-terminal domains. So we term these proteins mosaic, adhesive, trimeric proteins, and they really are. The sheer number of different domains we're seeing is quite incredible.